Thank you so much, Kim, for that introduction, and to the ASLO Organizing Committee, especially Roxanne and Jennifer, for planning such a wonderful week. It is such a tremendous honor to be this year's recipient of the Yentsch-Schindler Award, and also incredibly humbling. I'd like to begin by thanking my 3 lifelong mentors, Kathy Cottingham, Nelson Harston Jr., and Kathy Weathers, for their incredible support, guidance, and wisdom, and without whom I would not be here. These 3 mentors embody the legacy of Clerice Yentsch, for whom this award is partly named, and the critical role that mentors play in the continuity of science, and also for creating a scientific community in which we take care of each other. It truly does take a village. I've also learned from reading Clerice's work, as well as from my own mentors, the incredible privilege it is to be a mentor yourself. And for this, I've tried to pass on these lessons to my students, who have taught me far more than I could ever hope to teach them, and for whom I'm incredibly grateful. Thank you. As I was reflecting on this award, which is based on contributions to society, research, and teaching, and my own limnological journey, I felt that there were 3 take-home reflections that emerged that provided organizing principles for this talk. The first, the importance of building partnerships with community members, the public, and with managers. Second, developing and leading diverse interdisciplinary teams. And third, developing new teaching tools to train the next generation of aquatic scientists. It's really hard for me to separate out the society and research parts of my work, so I'm purposefully presenting those 2 together today. And this largely stems from my perspective that limnology serves as this perfect intersection between basic ecosystem ecology and applied management science. Within limnology, we ask questions about nutrient cycling and food webs, but the inherent end goal is to build our understanding of fisheries and drinking water. In turn, every time a manager makes a decision, that provides the rare opportunity for a whole ecosystem experiment, which ultimately will improve our understanding of how ecosystems function. It is through this lens in which I view my work as a knob that goes back and forth between basic and applied science that I began my career in 2004 when I contacted the Lake Sunapee Protective Association, or LSPA, which is one of the oldest lake associations in the northeastern US. I was looking for an undergraduate thesis project, and coincidentally that summer, Lake Sunapee, which is a large, deep, oligotrophic lake known for its very high water quality, began exhibiting a thick cyanobacterial scum covering the lake, similar as you can see here in the photo. The Lake Sunapee Protective Association asked me to investigate, which led to both an undergraduate project, but more importantly, my PhD. And this work, led with Kathy Cottingham and Kathy Weathers, I think has been really important for understanding and improving our understanding of how cyanobacterial blooms can not just be present, but can actually be common and may be increasing in low nutrient lakes across the northeastern US and Canada. We have learned that these blooms are prevalent, and we think that they're probably caused by a complex interaction between climate change, increasing storms, warming temperatures, as well as interacting with land use change occurring in many catchments across the Northeast. Importantly, once these taxa, especially my favorite, Gliotrichia, can appear in the water column, we think that it can accelerate nutrient cycling by unlocking sediment phosphorus and fixing nitrogen that can be released into the water column, thereby triggering other phytoplankton to bloom. This work was only made possible by a committed band of citizen scientists and community partners in the Lake Association, which provide us with data and field sites. And I have the honor of continuing to work with the Lake Sunapee Protective Association today, from my first annual meeting in the summer of 2005 to last summer. We're now working together on a couple natural human systems project, and we're studying how human decisions affect lake water quality, but in turn, how lake water quality can inspire people to act, such as by forming watershed groups like the Lake Sunapee Protective Association. During my PhD, I became deeply involved in GLEON, the Global Lake Ecological Observatory Network, to provide context for my work on Lake Sunapee as to how lakes around the globe are changing in response to human activities. GLEON is a grassroots network of scientists that are deploying instrumented buoys and sensors on lakes and reservoirs around the globe. GLEON provides a safe operating space for collaboration, thereby catalyzing novel freshwater science. The network has a very strong emphasis on early career training, from which I have benefited enormously. But I also think that GLEON's perhaps largest contribution is that it's enabling cross-site syntheses across gradients of land use, latitude, and many other factors, I think truly transforming limnology as a discipline. It is with my GLEON hat on that I arrived at Virginia Tech 5 years ago, and I immediately began developing a collaborative partnership with an amazing water utility, the Western Virginia Water Authority, to deploy instrumented buoys on drinking water reservoirs located around me in southwestern Virginia. Similar to my partnership with the Lake Sunapee Protective Association, the Water Authority has provided me amazing access to sampling sites and to data that I can use to ask and answer fundamental questions in limnology. But at the same time, I can also use those data to help provide drinking water management recommendations for the utility. As a result of this partnership, my research program expanded from studying phytoplankton blooms to now studying reservoir ecology and biogeochemistry. Reservoirs are incredible ecosystems, and we know so little about them relative to glacially formed north temperate lakes. Importantly, many reservoirs are heavily managed systems, and as I mentioned earlier, this provides the opportunity to do whole ecosystem experiments. The importance of whole ecosystem experiments in limnology stems largely from the legacy of David Schindler, for whom this award is also named, and I'm honored to walk in his footsteps in this regard. In my reservoirs, we have hypolimnetic oxygenation systems, in which we pump out the hypolimnion on shore, we inject oxygen at supersaturated concentrations relative to temperature, and then return that oxygenated water back into the hypolimnion without altering water temperature or thermal stratification. Thus, I can do whole ecosystem manipulations of hypolimnetic redox. I also have the good fortune of having upstream reference reservoirs that have the same land use and water chemistry, and thereby provide a reference for this oxygenated experimental system. I can thus turn on or off, let's see if I can do this, an oxygenation system, and then compare the ecosystem dynamics in the oxygenated reservoir referenced to when the oxygen is off, as well as in our reference system. Here on the x-axis is time, the y-axis is depth, and the color refers to the dissolved oxygen concentration. Thus, I can shift this ecosystem on a gradient from oxic to anoxic and in between and look at how coupled biogeochemical cycles respond. We've learned that when the reservoir is in its anoxic state, we have very high release from phosphorus and metals from the sediments into the water column. But when we shift the ecosystem to its oxic state, we find that metal and phosphorus internal loads are much smaller, but we have very high efflux of CO2, carbon dioxide, from the reservoir into the atmosphere, which makes sense because we're oxidizing organic matter that has accumulated in this reservoir's sediments for decades. Preliminary work in my lab suggests that the amount of CO2 that we're releasing may even offset the amount of methane that's now not being produced because of the oxygen in terms of global warming potentials. This work is of active research inquiry in my group, and I'm really excited about it. But I think that where we are right now is I want to note and just point out that we're creating these really interesting trade-offs between water quality and carbon burial as we move between this oxic and anoxic state. And this is important because we're increasingly experiencing variable oxygen conditions as a result of land use and climate change into the future. I'm now instantiating these whole ecosystem rates that we're measuring into ecosystem models that are fed by data from high-frequency sensors that are wirelessly streaming the data into the cloud. We're able to generate water quality forecasts, similar to weather forecasts, such that I can predict that on Thursday, there might be a 95% chance of an algal bloom. This is work that's being done in collaboration with a very interdisciplinary team of computer scientists, social scientists, engineers, and managers to study how managers might use these forecasts to make decisions about whether or not to preemptively treat the reservoir to prevent adverse water quality events from occurring. For example, if we know that an algal bloom might happen on Thursday, should we pump in more phosphorus today, or pump in more oxygen today, to prevent phosphorus release from the sediments? I think one of the most important parts of this work, though, is the social science component, because if we're not making forecasts that are useful for the managers, then there's really no point in developing the forecasting infrastructure and models in the first place. The last part of my limnological journey is thinking about how best to train the next generation of aquatic scientists. As part of my work and many others in Guyane, we are deploying high-frequency sensors in lakes and reservoirs across the globe, and collecting data on timescales that we previously never thought possible. This is an extremely exciting time to be a limnologist, but we're also creating a data deluge. And I think there's also a fundamental disconnect in why are we collecting all these data if we're not also developing teaching tools to train our students how to analyze and visualize and manipulate these data. In response to this challenge, my colleagues and I have developed many sets of teaching modules or flexible teaching materials that use real, messy sensor data sets to teach ecological concepts while also training students in data analysis skills. This is work that has emerged out of Project EDDI, or Environmental Data-Driven Inquiry and Exploration, and Macrosystems EDDI, two NSF-supported collaborations of education researchers and ecologists working together to develop flexible teaching materials that can be easily adapted for any classroom. I'm particularly excited about the next generation of this project, Macrosystems EDDI, which has developed a completely R-based curriculum to teach students lake ecosystem modeling. All of our materials are open access. I encourage you to check it out and let me know if you have any questions. To date, we have developed over 12 teaching modules, 6 of which use lake data from either Guyane or Neon. And these have been taught to over 10,000 students across the US and beyond. We have found from our formal assessment program that completion of an EDDI module of just a 3-hour lab period can significantly increase students' understanding of ecological concepts, such as how climate change is affecting lakes. But we've also found, perhaps more importantly, is that participation in the EDDI curriculum can significantly increase students' interest and confidence in working with big data and models. And this interest and confidence part to me is really important because they've both been linked to retaining underrepresented minorities in STEM. So I can only speak to my experience and my limnological journey. So I asked the broader question of how can we enable the success of all early career researchers within the ASLO community? Or in other words, what is the lifting tide that raises all boats? And I would argue that my take-home reflections may be applicable and useful to other early career researchers. For example, working with community members and the public has been incredibly personally rewarding to me and also enriching to see my work translated into actionable science. Similarly, working in diverse teams of computer scientists, education researchers, social scientists, and more has incredibly broadened my perspective, but it's also forced me to be a much better limnologist. And finally, developing new training tools to familiarize and get students working with big data and models will benefit them regardless of whatever career path they pursue, within limnology or beyond. But you'll notice if you look closely that there is a common theme to all 3 of these, and that's collaboration. Because it would simply not be possible for me to do this work without my fabulous collaborators and students who have supported me throughout and have pushed me to be the best scientist I could be. Given the challenges that our freshwaters are facing in terms of climate change and land use change and many more, I truly believe that we're in an all-hands-on-deck moment in the aquatic sciences. And it is only by collaborating and working together can we truly tackle and solve these challenges. So with that, I would like to thank the awards committee for this amazing honor, my family, mentors, lab, collaborators, and colleagues, and a special thank you to the National Science Foundation for generously supporting this work. Thank you.